Hello my dear friends, myself is Dr. Avinash Amangi. I am from Gurudev Academy, and last time we have discussed with respect to this particularly SSLC Model Question Paper-II and remaining whatever the questions which is related with respect to the biology part, mainly science, right? So we'll discuss one by one where we have actually left, right? So with respect to the questions, if you find out, already we have discussed all these aspects and we left out with respect to the this question, right? So we'll go ahead with respect to the discussion of this particular question. That what question says we'll understand first, right? A tall pea plant is crossed with a dwarf pea plant. Mention the types of a plant obtained in F1 generation and represent the result obtained in F2 generation with the help of what checkerboard, and mention the ratio of what varieties of particularly what plant, right? So overallly this is nothing but what the particularly Mendelian concept, and we are going to call it as what the monohybrid cross. Majorly what we call it as the monohybrid cross, right? Because why we called it as a monohybrid cross? Because here the only one character selected, that is nothing but what height. Height is a character, that is either it is tall or with respect to that the dwarf, right? So these are the what the height character what actually Mendel has selected, and then he carried out this particularly what monohybrid cross. With respect to the question what they are asking, the particularly the plant obtained in F1 generation, then they are asking it with respect to the what F2 generation, mention the what even ratio with respect to the what the particularly checkerboard. So we'll see how this particularly when tall pea plant is crossed with respect to the what dwarf tea plant, right? So dwarf pea plant, that what we are going to see. So what they mentioned with respect to the question is the tall plant which is actually crossed with respect to the what the dwarf, right? So here whatever the TT, capital TT, small t, what I am representing, that is known to be the what alleles. And what allele is pair of a gene, right? So that particularly it's a genotype of a particularly what organism. Now how it is like genotype and phenotype, that we will see here, right? So phenotype is what capital T is nothing but what it's a tall, right? Small t is nothing but what it's a dwarf, right? So this is what actually the question says, right? So with respect to that only, like this capital T or small t is nothing but what it is a genotype of an organism, right? Which is actually coding for these characters. Now genotype is nothing but it's a genetic makeup of phenotypic character. Now what is the phenotypic character here? That is nothing but what tall and dwarf. So I will call it as what phenotype, right? And the conditions what we have to see, this both of them are what identical alleles. So I am going to call it as homozygous, right? If these both of them are homozygous, then definitely the gamete formed will be of what only one type, that is capital T, right? Here also the gamete formed will be of what only one type, right? So I am going to call it as what gamete now. Now when these gametes get fuses or fertilization takes place from these both of the plants, that capital T, small t, right? Now when it becomes capital T, small t, this condition I am going to call it as, this condition is known to be the particularly heterozygous condition, right? So I am going to call this condition as heterozygous, right? With respect to the heterozygous, and even this generation I am going to call it as F1 generation. I'm going to call it as what? F1 generation. Now when it is a F1 generation and heterozygous type, so what we know is the tall is actually dominant over what dwarf. What Mendel obtained is tall is dominant over what dwarf. In F1 generation he got this particularly what the particularly allele which is of what non-identical. Why non-identical type? The gene is what non-identical here. This condition I call it as what homozygous. So it produced what only one type of what gametes. When they got fused, it became what heterozygous. So non-identical form of what alleles or genes what we are going to find out with respect to this particularly what heterozygous conditions. Now as tall is dominant over dwarf, then phenotype will be what here? The tall. So first questions what they have asked in F1 generation, what type of a plant they are going to obtain? They are going to obtain nothing but what the tall, because tall is dominant over what? Dwarf, right? So this is what the conditions what they have got. Then we'll discuss with respect to the what they even asked what F2 generation. This is completion of what F1 generation. Now what Mendel has done, he carried out selfing of this particularly what capital T small t, means this tall plant he taken into what F2 generation. In F2 generation he have taken this particularly what tall plant, right? So when capital T small t, this is one plant male consider, and this is capital T small t, this is one more plant which is nothing but what female consider. Now as the condition is heterozygous, with respect to the checkerboard if you consider, how we are going to draw it? Like this, right? Now as heterozygous means it can produce what two types of what gametes, right? So one I am going to represent it as male and one is what female, right? So with respect to the what female if you consider, capital T is one gamete, as the condition is what heterozygous, then small t is one more gamete, so two types of gametes produced by means of what whom? The female, right? Then you consider with respect to the male, the condition is again heterozygous, then what he obtained, that is capital T is one gamete and small t is one gamete. So like this what we are going to find out overall the particularly gametes, right? So there are what two types of gametes produced, because in F1 generation whatever the tall obtained is, that is nothing but what heterozygous. Why it is I am calling it as a tall? Because tall is dominant over what? Dwarf. Now as it is this F1 generation, when it is what self crossed, right, male and female plant, so there it is going to produce two types of what gametes, right? So this is what the representation of what checkerboard, right? So now when I am going to cross, first cross here, so what I am going to get? Capital T small t, right? Then take this t again and cross over with respect to this, what I am going to find out? Capital T small t, right? Then if I take this condition here, capital T small t, right? Then from here I am going to take this one, so capital small t, like this of what condition actually occurred, right? So gamete wise conditions if you consider overally, right? So that we will see how it is going to act on, right? So with respect to this if you consider, right, so this is the conditions what I have got, right? So one is what homozygous, two of them are what heterozygous, and one more is what homozygous. So with respect to that if I consider overally, this capital T will be what? The tall, right? This is the tall, right? Then capital T small t, what I have got into F1 generation is tall again, right? So heterozygous even I have got a tall. Then again the capital T small t is heterozygous tall, because tall is dominant over dwarf. So this is small t is what? The dwarf, right? Now with respect to the law, what it says, when a contrasting character are brought together, right, in a F1 generation, one of the allele dominates another by suppressing like what? Other gene, other allele, right? And they are going to get segregate or separate from each other during what? The formation of gamete in F2 generation. They got separated and segregated from what? Each other. Because of that only what we have got? The first generation, the first wave when we have crossed homozygous condition, the particularly what we are going to call it as wild variety, that even we have got over here. Now what this particularly phenotypic ratio and genotypic ratio is like? So phenotypic ratio and genotypic ratio if I consider, so what I am going to get? Consider first I will take phenotype. Phenotype is the character which you are going to see, right? Now what character I am seeing is tall, dwarf, right? So how many of them are tall? 3 of them are tall. So phenotypic ratio is 3:1, right? Then I am going to call it as what? Genotypic ratio. So genotype means what? The genetic makeup of a particularly what phenotypic character. Now, if I consider this tall, capital T, capital T, capital T, capital T, homozygous, homozygous form, how many of them are there? Only one. So I'll represent it as one, right? Then capital T, small t, how many of them are there? Heterozygous condition, two. Because this heterozygous condition, why it got arised? It arised due to the what? When it formed F1 generation, right? So only the ratio is 1 is to 2 is to 1. 1 is what? The dwarf one. Understanding? So the dwarf condition, what I am going to find out. So whatever the character was there into the previous generation, which maybe did not got expressed into the F1 generation, but that character I am going to get it into the what? The F2 generation. This is what we are going to call it as law of segregation or even law of purity of gametes. So with respect to the question, what they have asked, mention the particularly F1 generation plant. F1 generation plant is what? A tall, right? With respect to the what? Checkerboard, this is what overall is the checkerboard, and the ratios what they asked, one is phenotypic ratio and one more is what? Genotypic ratio. So phenotypic ratio is 3 is to 1 and a genotypic ratio is 1 is to 2 is to 1. So this is what the answer for that particular question, right? So one more question they have asked here, right? So first we'll try to understand what exactly question says here, right? So round green colored seed, right? Round green colored seed producing pea plant. So they have mentioned here what genotype, that is capital R and small y, right? What it is? It is nothing but what? Round green. So that even I am going to mention here, round green, right? Are crossed with wrinkled yellow colored seed producing a pea plant. That is what I am going to get here is small r, capital Y, right? So what it is? It is nothing but what? Wrinkled yellow, right? This is what the particularly I have with respect to the word questions. Then show the result of what? F2 generation with the help of what? Checkerboard, and mention the ratio of what? Varieties of plant. So overallly we have to find out the particularly checkerboard of it is. First what we are going to obtain? F1 generation. Then we are going to go for what? F2 generation. So you remember this round green, wrinkled yellow. In F1 generation, as you can consider both of them conditions. If I consider round, this is what? Homozygous. If it is homozygous, before only what I have explained, it is going to produce only one type of gamete. This is small y, this is also homozygous, it is going to produce one, right? So one type of one, what I am going to get over here is, right? Here wrinkled yellow, if I consider, here also what? This is wrinkle is what? The smaller, which is what? The homozygous. So I have got it what? Smaller. Then here this is also conditions yellow, which is what? The majorly what we are going to find out, the particularly only one type of gamete, homozygous condition, that is what? Y, right? So when these both of them are going to get what? Mating. So with respect to the mating, what I am going to get? Capital R, capital Y, small y, right? So capital R, small y, capital Y, small y. So what we know is this, whatever the character we have got, round green and wrinkled yellow with respect to the dihybrid cross, these of them are what? Non-parental combinations, what Mendel has got, right? So based on that only Mendel, what he says, when a pair of what? When two pair of contrasting characters are brought together, right? Neither brought together, one of them is going to be dominant, one of them is going to be recessive. In F1 generation, right? This is going to happen in what? In F1 generation. In F2 generation, the gametes are going to get separate, segregate, independent of what? Each other. That itself what we are going to call it as independent assortment. So whatever this we have got. So what they are asking lastly, F2 generation with the help of what? Checkerboard, and mention the what? Ratio, right? So again, this is what I have got it into what? F1 generation. Now when capital R, smaller, capital Y, small y, then what it will be? It will be round yellow. What it will be? The particularly round yellow, right? So this is what in F1 generation what I have got. Now overally with respect to the what checkerboard, what I am going to do, I am going to selfing this, right? So that selfing we will see like how it can be like, right? So what I have got into F1 generation, capital R, capital Y, small y, which is crossed with what? Capital R, capital Y, small y. What it is? It is nothing but round yellow, right? Here also what I have got is round yellow, right? Now with respect to that only, like consider orally what I am going to find out, the gamete with respect to the what checkerboard I am going to find out, right? So I will draw the particularly what checkerboard. Now what I am going to get, there are what the different types of what gametes, one is what female, one is what the male, right? So crossing based on that, what particularly first moment I am going to get it as the capital R, capital Y. Then one more type gamete is what small r, capital Y. Then I have got it what majorly capital R, small y. Then small r, small y, right? Again here also capital R, capital Y, right? Then small r, small y, capital Y. Then capital R, small y, small r, small y. So this is what the gamete what we have got, right? Because as is this condition is heterozygous one, right? So based on that only we are going to what solve this, right? So what I have got, capital R, capital R, capital Y, which is nothing but round yellow, right? Then capital R, capital Y. So this is even going to be round yellow, right? Then capital R, capital Y, small y. This is also going to be what? Round yellow. Because capital Y is dominant on what? Small y, right? Then what you are going to find out? Capital R, capital Y, small y. This is even going to be round yellow, right? Now what we have to remember over here is whichever the like what the dominant character is actually represented with respect to the what capital letter, right? And this is a dominant character, that what we have to remember, right? Then small r is or small letter is represented with respect to the what the recessive character, right? Here capital Y is dominant and the small y is what the recessive. This is what we have to remember over here, right? So based on that only what we are going to consider the particularly what characters. Again capital R, then small r, capital Y. Then what I have got is here again round yellow, right? Then small r, capital Y. This is what I have got. Now small r, capital Y. What it is? It is nothing but what? Wrinkle now. Wrinkle yellow. What it is? The particularly wrinkle yellow. So what I will do is I will mark it with respect to the what? Yellow. Right. This is one character what I have got. Right. So again if you consider capital R, small r, capital Y, small y. So again what I have got over here is the particularly round yellow, right? Then small r, capital Y, small y, right? So what I have got over here again the particularly wrinkle yellow. What I have got is the particularly wrinkle yellow, right? Now with respect to that again I am marking it with respect to the what? Yellow, right? Then next what I am going to get over here is small r, capital R, capital Y, small y, right? Then what I am going to get over here is round yellow, right? Then capital R, capital Y, small y. What I have got over here is the round yellow. Understanding? Then capital R, small y. What I have got is, so what it will be? It will be round green. What it will be? Round green. right? So that I'll mark with respect to the what? Green. So this will be the what? The green, right? Then next what will it will be? Capital R, small r, small y, right? Then what again got I have got over here is round green, right? So round green I have got. So this even I'll mark with respect to the what? The green. Understanding? Then next what I'm going to get over here is the capital R, capital Y, small y. This is even going to be what? Round yellow, right? Then small r, capital Y, small y. Now what it will be? It will be wrinkle yellow, right? What it will be? Wrinkle yellow, right? So again I'll mark it with respect to the what? Yellow, right? So this is what I have got. Then next what it is? Capital R, then small y. So what I have got over here is the particularly round green. What it is? Round green, right? So with respect to that, if you find out, the particularly this will be the green, right? And lastly what I'm going to get is small r, small y, which is nothing but majorly wrinkle green, which is a recessive character, right? So overall if you consider, this shows what three of them are what? Yellow, right? That is wrinkle yellow, non-parental combinations, what we have where we have actually crossed the first one, right? Then one more was what? Round green, that even what we have crossed. So overall this is what overall expression of what? The particularly Punnett square with respect to the what? F2 generation, right? With respect to the what? F2 generation. Then ratio wise if you consider, ratio wise how we are going to calculate? Like overall ratio if I will consider here, the phenotypic ratio what it is going to give me? 9 of them are what? The round yellow. See 1, 2, 3, 4, right? Then 5, then 6, right? 7, 8 and 9. So 9 of them are what? The particularly wrinkled yellow, right? Then what I am going to see is the particularly next is consider wrinkle yellow. Consider what we are going to consider wrinkle yellow. Wrinkle yellow if I calculate here 1, this is 2, right? And this is 3, right? Then ratio will be 9:3, right? Then again round green if you consider, that is even 3, right? And wrinkle yellow is lastly what? 1. So this will be the phenotypic ratio of what? 9:3:3:1, right? To calculate particularly what? The genotypic ratio, right? So now what you have to see, you have to actually find out the what type of combination. How many of them are with capital R, capital Y? How many of them are capital R, small r, capital Y? So like this of combinations what we have to find out. But I'll tell you the simple methodology to calculate what? The genotypic ratio. Right. Now what will be the ratio? How you are going to calculate in simple way? Like I'll erase all this, right? With respect to that genotypic ratio, what I have to calculate. Now what we know is in monohybrid cross, right? Monohybrid cross, what we have got? We have taken only one character. What we have taken? Only one character. Understanding? Now only one character basis, what was the particularly genotype of monohybrid cross? That what? Easy to remember, that is 1:2:2. Now whichever the which checkerboard we have solved, right? That was what? A dihybrid cross, right? So when it is a dihybrid cross, then based on that what I am going to do, I am going to multiply this is twice, right? 1:2:1. This is for one character. Now I am going to calculate for what? 2 characters. Then 1:2:1. I am going to calculate as 1:2:1. So with respect, if I am multiplying it, how I am going to multiply? You consider first you take this one, multiply this whole, right? Then next you take this 2, multiply this whole. Then next this one, multiply this whole, right? Now what will be the ratio? First moment you are going to get 1:2:1, right? Then second moment you are going to get 2:4:2:2. In last moment you are going to get 1:2:2:1. So this will be the actual genotype of what? The particularly the dihybrid cross. Understanding? And that was actual question for this particular answer for that particularly what question. The genotypic ratio and phenotypic ratio was with respect to the dihybrid crosses. That even we can calculate. How we are going to calculate? Consider the genotype of this phenotype of this particularly monohybrid cross. What it was? It was 3:1, right? So 2 characters, then I'm going to multiply it 3:1. So same way only, multiply this 3 with whole, then multiply this 1 with this whole, right? If I multiply 3:3, 9, right? Then 3:1, what it is? 3. Then 1:3, 3, and lastly 1:1, right? So based on that what multiplication even we can write it this particularly what phenotype as well as what genotype, right? So this is the simplest way what we have to remember in this particularly overallly either it may be monohybrid cross or dihybrid cross. The capital letters, majorly whatever the capital letters represented, that will be the dominant character. And the small letters, what it will be represented, that will be the what? Majorly the particularly recessive character. So capital letter is a dominant character and small letter is a recessive character. If you understand this, then there is then there will be a easy method to understand what type of a character or genotype or phenotype we are going to get it into the what? This different type of what? The generations, right? So we'll see the what next question, right? Now what this question says, the particularly draw the diagram showing the schematic section view of human heart labeled with respect to the following part. So we have to draw one particular structure, that is aorta and pulmonary veins, right? So aorta and pulmonary veins, that what we are going to show over here is, right? So this portion is nothing but called as what? Aorta. And even they asked is what? Pulmonary vein. This is what you are going to get it, pulmonary vein. Now what it is, like the particularly what the whatever the oxygenated blood, right? So that is going to move up with respect to the what? The blood from the heart to the what? Body part is moved with respect to the what? Aorta. From the body part the blood is going to come, that is nothing but what? With respect to the what? Pulmonary vein. So which is going to get enter into the what? Left atrium, left ventricle. Here from heart to what? Body part, from there where it is going to enter? Right atrium to the particularly right ventricle, right? So like that what the particularly movement what we are going to find out. Aorta and pulmonary veins, both of them are the particularly which is going to initiate the movement of this particularly what? Blood from heart to the part of what? The particularly other organs, or from the other organs to the what? Heart. So that what movement can be initiated by means of aorta and pulmonary vein. So they diagrammatically what they have asked over here, right? Then they asked one question, that is nothing but what are the tropic levels and follow of what energy in an ecosystem is always unidirectional. Why? That what we have to explain. What is actually tropic level, right? Now the tropic level of an organism is the number of what? Steps. It is from the start of the particularly what? Chain, right? So number of what? Steps basis only what we are going to understand here. So first primary, if you consider, the primary of them are what? The producers. Majorly we are going to call them as what? Plant, right? From the producer I am going to get what? The particularly primary consumers, right? Following to that I am going to get what? Secondary consumers. On to that I am going to find out what? Secondary consumers, tertiary consumers, right? So like that the particularly tropical level energy is going to flow in this particularly what? Tropics. Now who is going to trap up the energy from the sun? The producers. From the producers the primary consumers are going to consume, right? So here a food web starts with a what? A tropic level 1, where the primary producers are there, such as what? Whom? The particularly plant, right? Then the herbivores, the plant eating animals, that we are going to call it as primary consumers, and the secondary one is nothing but what, carnivores, right? So like that it is going to get what, arranged, and this is what we are going to call it as trophic levels, right? Now based on that only, the answer for this particularly what we are going to go, flow of energy is in an ecosystem is always what, unidirectional, and why, right? This energy, right, is then passed on from one organism to another in a what, food chain, right? One tropical level to another tropical level, if energy is passing, it means what, one organism to another only the energy is passing. The energy flow in the ecosystem is unidirectional. Why it is unidirectional? Because the energy lost as in the form of what, heat from the what, a living organism of a what, the particularly food chain cannot be reused by the plant in photosynthesis. Now what is going to happen here? We are going to calculate like majorly net primary productivity that is produced by means of plant, with respect to that what, gross primary productivity, and even respiratory loss or even heat loss, right? So which is going to give me the what, net primary productivity. Now plant is not going to utilize again that energy. Plant is going to give what, whatever the net primary productivity to the next tropical level. Now it is not going to use it back, right? So only what we are going to call it as, it is unidirectional. It is going to flow from one tropic to the next, but it is not going to come back again, right? So that is what actually the reason what you are going to find out. Then what is actually the ozone, what here has there, and what is the function of what, ozone layer? What are the cause of this particularly what, a duplication of what, ozone layer? That what we are going to find out the answer. Ozone, what it is? Ozone is a molecule containing what, 3 atoms of oxygen, right? That is O3, what we are going to call it as. Then a highly poisonous gas present into the what, upper layer of what, atmosphere, right? Then the formation of the ozone, how it is going to take place, right? Where what we are finding out here, the UV radiation splits the molecular oxygen O2 apart into the what, free oxygen atom, that O plus O, right? These atoms then combine with respect to the what, molecular oxygen to form what, ozone, right? What is going to happen? The particularly considered oxygen is there, then UV light is falling on it. What it is going to do? It is going to split this oxygen, right? Now it is going to get combined with respect to the one more oxygen to form something called as what, O3, right? So this is what actually the formation of what, ozone, right? So how it is going to take place in with respect to the what, particularly atmosphere, right? Then what they are saying is the particularly what is the function of what, ozone layer. Then function of ozone layer, if you consider, the ozone layer in the what, stratosphere absorbs what, it absorbs the portion of what, radiation from the sun, right? So sun radiations it is going to absorb, right, which actually preventing it from reaching to the what, plant's surface or planet's surface, earth's surface, before this particularly what, the high wavelength light or the short wavelength light which should not get entered into the what, earth's surface, which is to be what, absorbed by means of this particularly ozone layer, right? Most importantly, it absorbs what, the particularly portion of UV light, right, called as what, UVB, right? Then UVB is what kind of what, ultra light from the sun, and even that has what, several harmful effects, right? So this is what actual function of what, the particularly ozone layer. It is going to observe some specific wavelength of light. Then again what they are asking, what are the cause for the duplication of what, ozone layer, right? Now what will be the cause of what, duplication of ozone layer? The cause of what, ozone duplication is, ozone layer duplication is a major concern and is associated with a number of what, factors. The main causes responsible for the duplication of ozone layer are listed below, right? One is the chlorofluorocarbon, that is what we are going to call it as CFC, right? Then chlorofluorocarbons are the main cause of what, ozone layer duplication. So these are released by means of solvents, sprays, aerosols, and even what, refrigerator, air conditioner, from where the CFC is going to get what, release, which is going to affect something called as what, this ozone layer. the molecule of what chlorofluorocarbon in the stratosphere are broken down by ultraviolet radiations. Ultraviolet radiation, what it is going to do? It is going to break down this, right, and releases what? Chlorine atom, right, and these atoms react with respect to the what? Ozone and destroy it. Understanding? What is going to happen? UV light falls onto the chlorofluorocarbon and it releases something called as what? The chlorine, and that chlorine atom is going to react with whom? Ozone, and it is going to what? Destroy it, right. Then nitrogenous compound, consider the nitrogenous compounds such as what? NO2, then N2O are what? Highly responsible for the duplication of what? Ozone layer. Then again, some of the natural gases, if you consider, ozone layer has what? Have been found to be depleted by certain natural processes such as what? Sunspots and even stratospheric winds, consider, and it does not cause more than what? 1 to 2% of what? Ozone layer duplication. It causes less effect, but even it is a cause of what? Ozone duplication, right. Then volcanic eruptions, if you consider, are also responsible for the duplication of what? This particularly ozone layers, right. So this is what the major cause of what? These are the some cause of what? The ozone layer duplication, right. Then majorly what is actually reflex arc? Trace the what? Sequence of the event that occur in this particularly with respect to the structure when a bright light is focused on our eye, right, how our reflex is going to act on, right. Consider what this particular reflex arc is. This reflex arc is the pathway, right, that reflex takes place in the particularly what? Body. It's a pathway actually. Based on that we are going to carry out what? Reflex action, right. So reflex arcs function in order to what? Maintain the balanced and stable position, right. It is going to reflex arc functions in order to what? Maintain a balanced and stable positions. Reflex arc starts when a sensory receptor picks up external stimulation, and this information is transported by the what? Sensory neuron, which is also an afferent neuron, to the what? Spinal cord, right. How it is going to act on? It is going to start with respect to the what? Sensory receptors, which is going to pick up what? External stimuli, and with respect to that this information is what's transported by means of whom? The sensory neuron, which is also an afferent neuron, right. So to the what? Spinal cord. It is going to send to the spinal cord. From the spinal cord it is going to reach what? Brain, and then we'll have a what? Response against that particularly what? Stimuli. That overall whatever this procedure, that itself what I'm going to call it as the reflex arc, right. Then we'll see how this states the event of that occurs in with respect to the what? Bright light falling onto the what? Eye, right. Now consider when bright light, right, is focused on our eye, then it goes to the what? Brain, right. Bright light falls onto the eye, it goes to the what? Brain. The brain reverts back the message by a motor neuron, right, then which contract the pupil, which is going to contract the pupil. Then the sequence of event which occurs is, how it is going to occur? The receptor is going to take up the signal, pass on to the what? Sensory neuron to the what? Brain, from the brain to the what? Motor neuron to the what? Eye, and eye muscles get what? Contract. Majorly who is going to get contract? The pupil is going to get what? Contract, right. So schematically if you find out, this is what schematic you are going to find out. Stimulus with respect to the what? Bright light. Receptor eye, what it got, right, then it transferred it with respect to the what? Here the sensory nerves, which reached to the what? Brain. From the brain what? It is going to again go back to the what? The circular muscles of what? Iris, eye, right. Then the response is what? The pupil is going to get what? Shrink. So this is what actual schematic representation. You can pause the video and you can find out this particularly schematic representation of what? How actually we are going to respond with respect to the what? Bright light when it falls onto our eye, right. The next question is, that is, explain the structure and function of what? The particularly what? Placenta. Then what are the functions of prostate gland and the testosterone? One by one we will see. First we will see, explain the structure and function of this particularly placenta, right. Now what is this? The placenta enables, function of the placenta is what? It enables the exchange of oxygen and nutrient between the what? Bloodstream of blood stream of you and your baby without ever mixing them, right? So it is going to carry out what exchange of what blood, even exchange of what nutrient with respect to the mother to the baby, right? Now when there is an exchange, there will not be any what kind of a mixing, right? Then it act as your baby's lung. It is even going to act as what baby's lung, even kidney, liver, until the what particularly what birth, right? Then as you get closer to the delivery, the placenta passes antibody to your baby to jump starts its what immunity, right? So what it is going to do when it comes to the what delivery or the parturition, mainly what we are going to call it as, during that period the placenta is going to pass on what antibodies, so that baby even can resist the external environmental conditions, right? Then this is what overall the structure of what the particularly what placenta, right? So you find out what placenta surrounding to this particularly what baby, then baby is actually connected. This is well developed baby, you are going to find out connected with respect to the particular umbilical cord. Then overall this is what we call it as fetus mainly, then uterus and cervix. This is what actual placenta what we are going to find out, which is going to surround the particularly baby, help in what the exchange of oxygen, even exchange of what a nutrient, right? Then so with respect to that only, if you consider the function of what prostate gland, right? Second question it is, right? Then if you consider the prostate gland sits just below the what bladder, right? Prostate gland sits just below the what particularly bladder, right? Majorly if you consider, it surrounds the top part of the particularly tube, and that drains the urine from the what bladder, called as what urethra, right? Then the prostate main job is to make the what fluid that feeds the that is going to carries the sperm, called as what the seminal fluid, right? The prostate gland is a part of what male reproductive structure, right? Majorly what it is going to do, it is going to surround the particularly top part of this particularly drains of what urine from majorly what with respect to the what urinary bladder, right? So and called as what majorly urethra, right? Then the prostate main job is to what make the fluid, and that feeds and even carries what the particularly sperm. It is going to carry out the sperm with respect to the what the fluid is to altogether sperm and the fluid produced by means of this particular prostate gland, we are going to call it as seminal fluid, right? Or semen, even what we are going to call it as semen. Sperm and seminal fluid mixing is nothing but what semen. Then the function of what the particularly testosterone, right? So testosterone is involved in what the development of the male secondary sex characters, right? So male secondary sex characters or testosterone is known to be the what male hormone. So it is going to develop what the particularly secondary sex characteristics, such as what facial hair, the what we are going to find out the beard and all, right? Then pubic hairs, right? Increased what muscular build, muscle build, even what you can find out with respect to this particularly testosterone. Then the deep voice, right? It is involved in what increasing the particularly protein synthesis in even muscles, thereby increasing the muscle mass with respect to the what male, right? So testosterone is going to help up with respect to the what giving us a secondary characters, majorly with respect to the what males, because it's a male hormone produced by means of what the testis, right? So overallly this is what the particularly model question paper 2 and majorly biology part, right? So after watching watch this video, carry out your revisions and do well, prepare well for your upcoming SSLC examination. All the best guys, and do well with respect to the what even your examination. And most importantly, please do subscribe our channel, like and share even. Thank you so much.